In Essential Task 8.7, we are going to detail the historical and contemporary methods of researching the brain. I think this is a horizontal concept map. As you can see here, there are 5 columns from left to right, all under the broad classification of studying the brain. I'll return now to the task text, allow you to copy down the portion in yellow before proceeding into the concept map. So in Essential Task 8.7, we are going to look at the variety of ways that we study the brain and some of the methods that we've used to make statements and determinations about brain region and brain function throughout the unit. One of the first pretty unique methods of studying the brain is acknowledging a phenomenon called brain plasticity. Now, I need to make it very clear before talking about brain plasticity that this is much more likely when you are younger, when your brain is still developing. It is way less likely to achieve the same degree of success in your 20s or 30s, or certainly much older than that. So brain plasticity is a phenomenon that is more likely to occur when younger, but under some unique circumstances, there appears to be the ability of some neural connections to rewire or reconnect in the aftermath of some type of injury in order to achieve function that was lost. So I would like you to watch the entirety of this clip below. It's really fascinating. It's a young girl who literally lost half of her brain. In order to relieve epileptic seizures, they conducted what's called a hemispherectomy, which is to literally cut one entire hemisphere out. This young girl has literally just one side of her brain. However, her injury or her epilepsy was identified so young that her brain was able to essentially rewire itself and regain some of the function that was lost. In theory, if we cut the left side of your brain out, or let's say your right side of the brain, let's just say right side for this example. If we cut the right side of your brain out, in theory, there would be nothing to control the left side of your body. As we'll see in a box upcoming, the right side of your brain, for example, the right motor cortex, controls your left hand. Well, if we chop or cut off the right hemisphere, in theory, there would be no ability to move the left side of your body. And for a brief period of time, that might have been the case. However, due to brain plasticity and due to the young age, the child in this clip was actually able to regain that movement. So this phenomenon called brain plasticity is more apparent at a younger age, but it's a really tremendous example of some of the unbelievable things that the brain can achieve. Hemisphere specialization was the primary topic of focus by key psychologist Michael Gazzaniga. There's a typo here, it should say A-E-L, not Michel Gazzaniga, it's Michael. Gazzaniga conducted research on split brain patients. We briefly referenced the idea of split brain patients back in 8.5b when we talked about the corpus callosum. That was the region of the brain and the bundle of neurons that attached your left and right hemispheres together. Well, patients, in order to alleviate epileptic seizures, who have had their corpus callosum severed are what we classify as split brain patients. And these unique conditions allow us to make determinations about what skills are localized in one hemisphere versus the other. Because for 99+% of people, information in the left side of the brain goes to the right side of the brain and vice versa, because for most people, your corpus callosum is intact. For individuals, however, who have their corpus callosum severed as a medical procedure to alleviate epilepsy, what happens in the left side of the brain doesn't necessarily go to the right side of the brain. So if there's a specific skill that the left side or specific bit of information that the left side possesses, it would in theory be unable to inform the right side. And if there are unique behaviors and skills that only one side of the brain can do, that's going to create some very interesting studies or setups that we can talk about. I would like you to watch the entirety of this video here. In order to really understand what is about to happen, you need to watch the entirety of this video here. But I'm assuming that you've watched that video, and I'm going to kind of explain what you saw in that clip. And it's a little confusing. I completely get it. We're going to do a lot of flip-flopping here. Luckily, in class, we're going to work on this. So if this seems confusing in the context of a YouTube video, I totally get it. But information in your left visual field, which isn't exactly how it works, but let's just say what your left eye picks up, but your left visual field. Information in your left visual field is flipped and sent to the right side of your brain, meaning it goes to the right occipital lobe. Information in the right line of vision, or the right field of vision, is sent to your left occipital lobe. So the line of sight or the field of vision, left versus right, flips to the occipital lobe. Left field goes to the right occipital lobe, right field goes to the left occipital lobe. That's step one. Your right hemisphere controls the movement of the left side of your body. Your left hemisphere controls the right side of the body. I know we're flipping around a lot. The left hemisphere, as we'll see in just a moment, contains Broca's area and Wernicke's area. As we established in 8.5b, those are the regions of the brain responsible for language comprehension and the production of language. Meaning, if I flash the word face to the right field of vision, it's going to go to your left hemisphere. For 99% of people, plus, not more than 99% of people, if I were to do that, you would be able to say face out loud. Why? That information reaches your left hemisphere. That's where Broca's area is. You can say the word face. If I were to sever your corpus callosum, though, and this is where it might seem a little weird, but hopefully you watch the video and you'll understand this. If the left and right sides of the brain don't communicate with each other, it creates this interesting phenomenon where, all right, well, what happens if I flash the word face to your left field of vision? If the word face is projected to your left field of vision, it gets processed in the right occipital lobe. There's nothing on the right side of your brain that can produce language, as we'll see Broca's and Wernicke's areas on the left side of the brain. For most people, information in the right side of the brain can be sent to the left side of the brain via the corpus callosum, and then you can say the word face. It happens instantaneously. Don't even notice it, no concern. But if we were to sever the corpus callosum so that your left brain and right brain do not communicate with each other, if information is sent only to the right side of the brain, so the word face is only being processed on the right side of your brain, there's nothing on that side of the brain that can produce the word face. You cannot activate the facial muscles necessary to say the word face. So you're actually going to see an individual who cannot say the word face. It's blatantly there in front of them. But because their corpus callosum has been severed, the left and right sides of the brain do not communicate with each other. We call this condition split brain patience, and it's a really unique setup that has allowed us to make some determinations about what the left and right side of the brain can do. And I like to stress you don't necessarily have to write this, though they have asked this on the AP exam in a context before. There's really no such thing as left versus right brained. What's way more accurate is to specifically denote function that appears present in the left hemisphere versus the right hemisphere. That's a much more accurate and adequate way of describing what the hemispheres do. The left hemisphere is language-based. We have Wernicke and Broca's area only in the left hemisphere, which is why those split brain patients, when the information was only in the right hemisphere, could not say the words. Logic appears to be a left hemisphere behavior as well. This is why a lot of people, when they talk about left versus right brain, say left brain is very analytical. Well, technically logic is founded there, but I think that's a little bit of an oversimplification. And then the left hemisphere controls the right side of your body. So it controls the right side's movement and the right side's touch. Both of those are localized in the left hemisphere. So the left motor cortex moves the right side of the body, and the left somatosensory cortex processes touch in the right side of the body. The right hemisphere on the other side seems to be more active during spatial or visual tasks, which is why people typically say the right brain is for artistic ability. A little bit of an oversimplification too. But for example, if you were walking through a maze, your right hemisphere would be going crazy. If, for example, you needed to navigate your way around a mall that you've never been in, the right hemisphere is clearly going to be active. And then it does the vice versa in terms of movement and touch. It controls the left side of the body's movement and the left side of the body's sense of touch. And really, the conclusions that you see on this table are a byproduct of studies like the one that Mike Gazzaniga did with split brain patients. His work, and clearly watching all the way through to the artistic elements at the end, really allowed us to make determinations about what each side of the brain does. The third box in is just to acknowledge a term that you could see on the AP exam. It's called lesion. It's typically not going to be asked, what is a lesion? But you could see a question where Bob's temporal lobe is lesioned. What might you anticipate in regards to his behavior? Well, that question would require you to have at least a general understanding of the word lesion. It means for there to be brain damage. And as a result of this damage, we can then make determinations about what functionality has been lost as a result of that damage. So this term would likely just come up in a multiple choice context, as opposed to it being what is a lesion as a direct question. The last part of today is to talk about the different manners in which we conduct brain imaging. There are two different categories, structure and function, meaning brain imaging techniques that denote structure basically give you a snapshot of what your brain looks like at any given moment, And you can typically use this to determine if there's been any traumatic brain injuries, the potential of there being tumors or other type of structural problems. It doesn't show you the brain actively working, not at all. It just shows you what the brain looks like. And it shows whether or not there's any apparent damage, again, regarding whether there could be tumors or some type of traumatic brain injury. And it's really great. You look at a CAT scan or an MRI, it looks like this. You see your cerebellum back here. You can see your pons, your medulla. Over here would be the corpus callosum. You can see all of the 4 lobes. So you really get a nice snapshot to ensure that the brain structurally is intact. There are 2 primary structural imaging techniques that exist, CAT scans and MRIs. I would like you to watch the CAT scan video here at the top. It's only about a minute or so long, but it gives you a nice snapshot into the functionality of what these 2 types of research methods entail. The other way that we can study the brain by looking at an image is to use what's called functional imaging. Functional imaging shows you what the brain looks like during certain tasks. So, for example, an fMRI, a functional magnetic resonance imaging study or imaging technique, a functional magnetic resonance imaging scan allows you to look at brain function because it will produce multiple MRIs back to back to back, and then you can watch changes according to each of the videos that you get. An fMRI, a functional magnetic resonance imaging, is a method where you take multiple MRIs back to back to back, and what it allows you to do is to see if there is any change from one scan to another. An individual might do, for example, a language task, and you can find areas of the brain that are active during language tasks. You see here, yeah, there's activity all over the brain, but there's a lot of activity on the left side in some of these, helping us, indicate, for example, this would be Wernicke's area, Broca's areas over here. There's a ton of activity there in Broca's area. So depending on what type of language task you're doing, we can use the fMRIs to denote activity. That's also true with PET scans. With PET scans, what you do is you inject radioactive glucose into the body of the patient. And when they are, for example, activating a part of their brain, when they're hearing or seeing something or speaking, it flares up on the PET scan machine. So you actually can identify the precise locations of some of the key behaviors. And if you take a look here, look, the temporal lobes are going crazy when you're hearing. Why? The neurons there are activating, and those neural impulses and the electrical charges that they produce triggered the glucose on the screen to light up, and you can actually denote precisely where some of this activity is. And then finally, there's an EEG. EEG is the most common method of studying the brain. It shows overall brain activity by scanning over the course of the night. Often you'll wear this device over your scalp, and over the course of your night's sleep, it will denote brain activity. It will allow us to determine what stage of sleep you're in and if there are any sleep disturbances. Please watch the entirety of this clip here at the top. It will help preview the sleep portion of this unit that we'll talk about later on. As a mnemonic, I look at the double E in EEG and the double E in sleep, and that's how I connect EEGs, electroencephalograms, to being the primary method of studying sleep.